Today we are going to discuss DNA and its structure. The DNA stands for deoxyribonucleic acid. It is composed of two strands that are coiled together to form a characteristic double helix. Each single strand of DNA is basically a chain of four types of nucleotides having the bases adenine, cytosine, guanine, and thymine. A nucleotide may be mono, di, or triphosphate deoxyribonucleoside depending upon one, two, or three phosphate attached to a deoxyribose sugar. The chemical interaction of these nucleotides form a phosphodiester linkage, creating the phosphate deoxyribose backbone of the DNA double helix with the bases pointing inward. This is the structure of the nucleotide. The nucleotide is composed of nitrogenous base that is attached to the carbon number 1 of this pentose sugar, and a phosphate group that is attached to the carbon number 5 of the pentose sugar. Now the DNA backbone. This is the DNA backbone. Here we see that the phosphate is attached to the carbon number 5 and the bases are attached to the carbon number 1 of the pentose sugar. There exists a phosphodiester linkage between the two deoxyribose sugar. For example, if the phosphate of the one nucleotide form a phosphodiester linkage with the hydroxyl of the second nucleotide, the hydroxyl is present on the carbon number 3 of the another sugar. This is the simplified structure of the DNA. Here we see that the guanine is paired with the cytosine and the thymine is paired with the adenine. There are 2 hydrogen bonds present between thymine and adenine, while 3 hydrogen bonds are present between guanine and cytosine. The DNA strand have directionality, and the different ends of a single strand are called 3' end and 5' end, with the direction of naming going from 5' to the 3' region. The strands of the helix are antiparallel, with one being 5' to the 3, then the opposite strand 3' to 5'. For example, here we see that if one of the strand is 5, the opposite strand, if the first strand start from the 5' end, the opposite strand will start from the 3' end. If this strand is 5' to 3', the opposing strand will be from 3 to 5, 5 to 3, 3 to 5. The term prime refers to the carbon atom in the deoxyribose to which the next phosphate in the chain attaches. The directionality has a consequences in the DNA synthesis because the DNA polymerase enzyme that is necessary for the synthesis of DNA will add nucleotide to the 3' of a DNA strand. The pairing of bases in DNA through hydrogen bonding mean that the information contained within each strand is not unnecessary. If the nucleotides on the single strand are present, these nucleotides can be used to reconstruct the nucleotide of the second strand, or we can say that the nucleotide on a single strand can be used to reconstruct nucleotide on a newly synthesized partner strand. Thank you for watching.